Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Unlocking CRISPR Advances in Base Editing, Prime Editing, and Future Applications. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank GenScript who developed the content for this presentation. GenScript is the leading contract research organization in the world providing gene, peptide, protein, CRISPR, and antibody. Since its foundation in 2002, GenScript has grown exponentially through partnerships with scientists conducting fundamental life science research, translational biomedical research, and early-stage pharmaceutical development. The company is recognized as having built a best-in-class capacity and capability for biological research services encompassing gene synthesis, peptide synthesis, custom antibody and protein engineering, and in vitro and in vivo pharmacology, all with the goal to make research easy. Now it's my pleasure to introduce our speaker for today's webinar, and she is Dr. Jiabo Liu. She is a dedicated senior protein scientist at GenScript with over 2 years of experience, and there she is, hi Jiabo, 2 years of experience in advancing the production processes for base editors and prime editors. She completed her PhD in biochemistry and molecular biology at Jylland University in 2022 and gained additional research experience as a visiting PhD student at Aarhus University in Denmark. Jiabo has authored 5 SCI papers in respected journals focusing on enzyme immobilization, semi-rational enzyme design, and prebiotic biosynthesis. At GenScript, her work is primarily centered on developing enzyme products and activity assays, particularly with CRISPR-Cas nucleases and molecular enzymes. Jiabo has also provided expert protein production and assay services to a wide range of organizations, including major pharmaceutical companies, research institutions, and universities, such as Merck, AstraZeneca, and the Broad Institute, I should say. Now, with the introductions out of the way, it is now my pleasure to pass the mic and the controls over to Jiabo. So Jiabo, when you're ready, you may begin. Perfect. We can see your slides.
Great. Thank you everyone for coming today. I am Jiabo Liu, Senior Scientist of E. coli recombinant protein production team from GenScript. I'm excited to be here today to share insights on the development of base editing and prime editing technology and its applications in gene editing therapies. In recent years, CRISPR-Cas technology has gained immense attention for its efficient and precise gene editing capabilities. From its initial discovery to its current clinical applications, CRISPR-Cas9 has undergone a soaring evolution. To start, the origin of CRISPR-Cas9 can be traced back to the bacterial immune system. Scientists gradually recognized that the system could be engineered into a powerful gene editing tool. in 2012, Jennifer Doudna and Emmanuel Charpentier first harnessed its technology as a gene editing tool, marking the beginning of a new era in genome editing. Starting in 2016, the concept of base editing was introduced, marking a major breakthrough in the field of gene editing. Unlike traditional CRISPR-Cas9, which relies on double-strand breaks in DNA, base editors enable precise single-base conversions in DNA sequences without cutting both strands of the DNA. This development made gene editing safer and more efficient, especially for correcting single-point mutations responsible for many genetic disorders. Since its introduction, base editing was rapidly evolved, with multiple versions developed to improve efficiency, specificity, and versatility, opening up new possibilities in therapeutic gene editing. Beginning in 2019, prime editing emerged as a powerful advancement in the gene editing field, developed to offer even greater precision than traditional CRISPR-Cas9. And base editing, prime editing, enables direct rewriting of specific DNA sequences without creating double-strand breaks. This technique was developed by David Liu's team in MIT, allowing targeted insertion, deletions, and base-to-base conversions. Since its introduction, prime editing has been refined for improved accuracy, versatility, and efficiency, holding tremendous potential for correcting a wider range of genetic mutations and advancing therapeutic applications. The first gene editing treatment, Gastly, was developed by Vertex Pharmaceuticals and CRISPR Therapeutics, who were the first to bring it to regulator for approval. This treatment is designed for sickle cell disease. However, it comes with a high price tag, expected to be between $2 and $3 million. There are numerous clinical trials underway as we speak. And I believe CRISPR-Cas technology holds the key to unlocking a healthier future for all living beings. Today, I'll be introducing base editing and prime editing, along with sharing some insights on protein production and its applications. First, let's review the gene editing mechanism of Cas9. The Cas9 protein complex contains 6 domains. The REC1 is the major domain responsible for binding with the gRNA, and REC2 function is not studied yet. The origin-rich bridge helix initiates cleavage activity upon binding to targeted sequences. Then the interaction with PAM confers PAM specificity, which is responsible for binding with the target sequence. The HNH and ROBC domain are nuclease domains to chop the target sequence. The Cas9 protein remains inactive due to the absence of gRNA. The programmed gRNA binds to the Cas9 and generates changes in the protein, which leads the inactive Cas9 protein into its active form. Once triggered, it searches the target sequence by binding with a sequence that matches the PAM sequence, which is NGG for Cas9. Then Cas9 generates double-strand breaks, DSB, at 3 p.m. upstream of the PAM using its HNH and ROBC domains. However, double-strand breaks can lead to serious consequences, making them less safer for clinical use. Single-strand breaks, SSB, on the other hand, provide a more reliable and safer approach for gene editing. So we use N-Cas9 as a nickase, so which will provide a nick on the double-strand DNA of the template. There are two N-Cas9, which is D10A and H8448. These two are different kind of N-Cas9. So building on the principle of single-strand breaks, base editing emerged as a groundbreaking technique in gene editing. This approach allows researchers to make precise alterations to DNA at specific base pairs without creating double-strand breaks, which can lead to unintended consequences. There are two types of base editors: cytosine base editors and adenine base editors. This scheme shows their editing mechanisms. Cytosine-based editors catalyze the deaminase of cytosine to generate uracil through the cell's endogenous mismatch repair pathways. Uracil is subsequently converted to diamine, resulting in a C to T substitution. CVEs often include a uracil DNA glycolase inhibitor, short for UGI domain, to inhibit cellular detection and correction of uracil mismatches back to cytosine, ensuring stable editing outcomes. Adenine-based editors utilize adenosine deaminase to convert adenosine to inosine, which behaves like a guanine during DNA replication, resulting in the targeted A to G base pair change. So from these two mechanisms, we can achieve C to G and C to T and A to G substitutions in the it's like a base to base editing. Here I put the common fusion protein architecture for CBE and ABE. For CBE, the commonly used cytosine deaminase is AppleBack1. There are also different family of AppleBack, AppleBack2, 3, something like that. And for ABE, the commonly used adenosine deaminase is TADA. I also get some requirement to screening different deaminase to create or to evolve better base editing efficiency. The first cytosine base editor is coupling cytidine deaminase with the inactive D-Cas9. They didn't use an Cas9 at the beginning. And fusing an inhibitor of uracil DNA glycolase to D-Cas9 prevents base excitation repair, which changes the U back to C mutation. So that's why that's how they find the need to fusion a UGI to increase the efficiency. And then N-Cas9 fusing cytidine deaminase and UGI was called as BE3. And a nick on the edited strain will be recognized as the DNA repair template. Over 60% editing frequency in human cells, and the indel frequency is only 1.1% of BE3. Next, I will introduce prime editing. First, prime editing requires an N-Cas9 and a reverse transcriptase. after Cas9 creates a nick on the template, the PEG RNA, which is this one, pairs with the template. And under the action of reverse transcriptase, a reverse transcription reaction occurs, allowing for the insertion and editing of longer segments. In addition to the nicking function of Cas9, the function of reverse transcriptase is even more crucial for implementing this editing method. Therefore, to maximize the efficiency, the reverse transcriptase enzyme should possess the following optimal characteristics: highly catalytic efficiency at elevated temperatures, ability to synthesize long cDNA products, high sensitivity, fidelity in DNA synthesis, effective bypass of DNA lesions, RNase activity, and strand transfer and strand displacement activities. These are the characteristics that our scientists right now and in the future will working on this PE system. And the reverse transcriptase commonly used in prime editing are as follows. The first one is from this virus, which we normally called MMLV. And the other one is the reverse transcriptase from AMV, and the reverse transcriptase from HIV1, and even some reverse transcriptase from thermostable bacteria. Therefore, improving the following properties of reverse transcriptase is essential for enhancing editing efficiency, which is also a major focus of research for many scientists today. Next, let's go over how PE performs gene editing. The PE editor is formed by fusing Cas9 and a reverse transcriptase and is guided by PEG RNA or prime editing guide RNA, which is PEG RNA, which is prime editing guide RNA. The PEG RNA is longer than a general SG RNA because it carries a reverse transcription template. And first, Cas9 recognize the PAM sequence and binds to the template, unwinding the target region of DNA helix for editing. Then Cas9 acts like a nickase, creating a nick. Creating a nick on the template. And after they create a nick, the prime binding site, which is also known as PBS, on the PEG RNA binds with the nicked template. So this prime binding site is also why we called this editing mechanism is prime editing. Using this section of the template as a primer and the PEG RNA as the template, a reverse transcription reaction occurs. Then inserting and will insert the desired sequence for editing. So the final results can lead to two scenarios. In one, the unedited sequence becomes the flap, while in the other hand, the edited sequence becomes the flap. This results. So this will result in two different final products. One is successfully edited sequence, and the other one is unedited sequence. So as we can see, This results in only a 50% probability of successful editing in the final PE products, even when disregarding factors such as PEG RNA design and deliver efficiency. To address this issue, scientists first engineered MMLV to enhance the stability of the protein and its nucleic acid affinity. We refer to this as PE2. Building on PE2, an sgRNA is added at a distance of 40 to 90 base pairs from the nick position. And then the sgRNA will combine with the Cas9 and make a nick. The cell's endogenous mismatch repair mechanisms will expand this insertion, remove the mismatched bases on the B strand compared to the A strand, leaving behind the B strand and leaving behind the A strand. The cells itself repair process will then generate our successful edit double-strand DNA. And the editing frequency can up to 33% with PE3. So after PE3, there is another architecture was developed by David Liu's team, which is called PE-Max. This is an architecture that they did two site mutations on Cas9 and the codon optimization of MMLV reverse transcriptase. Besides that, they also put NRS SV40 in the both end of this sequence and an NRS sequence from MAC at the C terminal of this sequence. What's more is a linker. SSGS is also important for this for stable the protein. So basically, to my understanding, this is a very useful structure, and maybe we can generate more or engineering the enzyme based on this architecture. After implementing these improvements, the efficiency of prime editors increased by 2.5-fold in HeLa cells and by 1.2-fold in HEC293 T cells compared to PE2. On this basis, scientists discovered a self-repair pathway called MMR, mismatch repair, that affects gene editing efficiency. Therefore, David Liu's team further incorporated MLH1 dominant to inhibit the MMR pathway, which will be introduced which will be used with PE2, and then this system would be called PE4. And based on PE4, if we use the strategy of PE3 by adding another sgRNA into the system, we will have another prime editor, which is called PE5. Therefore, David Liu's team further incorporates MLH1 dominant to inhibit the MMR pathway, significantly enhancing editing efficiency. So, besides engineering the enzyme and the reaction system, PEG RNA is also a very important factor in the prime editing system. So to stabilize the pegRNA in the system, we need the scientists notice the pegRNA is normally digested from the 3' end by the enzymes inside the cells. So a tetrary structure was added on the 3' end of the pegRNA, which will stable the pegRNA in the system. And this this is called engineered pegRNA, which is short for ePegRNA. By adding ePegRNA in the system, it will further improve the efficiency. And I think using ePegRNA in our different system like PEMAX system or PE4 or PE5 system, which will lead to maybe more surprising results in the future. And then in this project, the research team developed a PACE system designed to perform PE edits on defective phage genes, enabling Darwin selection of the phages. Over several hundred hours, phages carrying the original PE editor went through thousands of generations, rapidly evolving reverse transcriptase with entirely new structures. As a result, David Liu's team developed a series of new PE editors named P6A to P6G. Each system features a new reverse transcriptase or Cas9 variant. The team tested these PE editors on different types of gene editing tasks, discovering that P6A to P6G showed distinct editing preference. They improved editing efficiency in specific tasks, such as inserting short DNA segments, but were less effective in other scenarios. And some of the protein was the size of some of the PLUA to PLUG is decreased a lot, which will be benefit for the protein delivery in the clinical use. On April 3, 2024, Nature published the latest research finding from the team of Brett Edmondson and Lance R. Parsons at Princeton University, which is called Improving Prime Editing with an Endogenous Small RNA Binding Protein. In this study, the authors developed an editing system named PE7, which improves editing efficiency compared to the currently most advanced PE5 system. Through genome-wide CRISPR interference screening, the authors discovered that a small RNA binding protein, LA, is a key regulatory factor for PE efficiency. The LA protein and its truncated version, LA1 to 194, can interact with the poly-U at the 3' end of pegRNA, affecting pegRNA stability and thereby enhancing gene editing efficiency. Thus, it makes me thinking that the different using this fusion this LA protein or the truncated version with different PE system may lead to better editing efficiency in the future. Gene editing has numerous applications across various fields. I have served clients who ordered ABE8E for research on sickle cell disease, as well as clients interested in gene editing of plant cells. I believe As we all know, the CRISPR-Cas system can be used in plant biotechnology, food industry, biofoods production, drug discovery, development, and screens, of course, and control of bacteria and viral diseases, and correction of genetic and metabolic diseases, and livestock improvement. I believe that in the future, as gene editing technologies continue to mature, people's lives will improve significantly. And this is useful information that I prepared for you. I think different Cas system might use will use different palm sequence. So if you need to screen different Cas system, you can use this toolbox and to check what kind of palm sequence you are gonna use. When we are talking about CRISPR system, it is hard to talking about it's hard to not talking about the vectors for the in vivo delivery of CRISPR system. As we already know, there are a lot of different delivery system, but a lot of them was researched by delivering plasmids and the protein, delivering protein in the living cells or in the animals is still a very challenging project. Our GenScript is trying to produce high quality proteins to accelerating the research of protein delivery in the CRISPR-Cas system. So I have some case study that we have done for the different CRISPR-Cas editing system. As you can see, this one is we did produce ABE with a very good purity, which is over 90% purity by SDS page. And we also successfully produced the CVE protein in the E. coli system. And we also produced this PE system, PE protein, and in the E. coli system. And we all achieved over 90% by SDS page. The more detailed case study I'm going to show is this PE max protein, which is very common used in all the PE researches. As you can see, the purity of our protein is over 90% purity by SDS page and over 99% purity by SEC-HPLC. We also have very strict QCs to release our products. So we will achieve a concentration around 10 mg per ml, which will benefit for the electron transfer to the cells. And then our purity would be higher than 90% by SDS page. And the endotoxin level needs less than 10 EU per meg, which is suitable for cell treatment or even in injection into the animals. And our SEC-HPLC purity would over 99%. And there would be no specific DNase activity and exonuclease because some of our customer would like to try the in vitro activity or screening their pegRNA for their prime research. So they need to make sure the enzyme is nuclease free in case they digest their substrates. And we also successfully provided Med7 to our customer, and the QC of all our Cas protein was similar, and the concentration is all around 10 mg per ml and 90% purity by CS page. The endotoxin level needs to less than 10 EU per mg. The SEC HPLC results will be 89%, which will be vary from different proteins. But our criteria release level is over 95% purity by SEC HPLC, and non-specific DNA activity and exonuclease. We also did some in vitro activity of CRISPR-Cas protein, and this result shows that we tried to test their cleavage activity in vitro, and we produced the Cas12a we produced compared to another commercial Cas12a. It shows a better cleavage activity. As you may notice, there are less substrates left in our lines, but in the commercial Cas12a, you can notice there are more substrates left. So these days we have launched the guaranteed Cas protein nuclease package, and for all the Cas nuclease and base editor prime editors, we will provide high QC standard, and we will deliver all the protein from 2 weeks to 6 weeks. It depends on the sequence of the nuclease that you want. And our product like a delivery format would be comparable in release criteria. Package price included endotoxin control and nuclease removal. Also, we have small amount range for activity assay and screening friendly to enter try. And these are some existing protein package and QC results. We already have P2, Pmax, without RSH activity, and P6B, P6C, P6D, and Med7 in stock. We will provide trial size to the customers. And the QCs are in this table, which are the same as we produced before. And these are the clients that we have provided our protein to them. Especially for this CRISPR therapeutics, they give us very good feedbacks on our PE2 and PE max. And they tested the editing efficiency in the HEC293 cells, and it shows relatively comparable results to the literature. Okay, that's all of my slides. So back to Sonia.
Well, thank you very much, Jabo, for that great presentation. I hope everyone enjoyed it. Now, before we get into the Q&A portion of the webinar, we do have 2 simple poll questions for our audience members to engage with. I'm going to launch it now. So this is our first poll question. This is done in real time, so your participation is strongly encouraged and very much appreciated. And the question that we have for you is, what maximum level of the endotoxin control for a CRISPR-Cas protein in cell-based experiment? So please go ahead and make your selection from the below. Is it less than 100 EU milligrams, less than 10, less than 1, or is it less than 0.5? So please go ahead and make your selection. I'll leave this open for another 20 seconds to get everybody involved in this poll. And then once I see that we've got majority of our audience, I'll close the poll and take a look at the results and then get into poll question number 2. So please go ahead and cast your votes. Okay, that looks like it's the majority of our audience. So I am now going to close up. Okay, hold on. I'll leave it open for a few more seconds. There's a few more people making their selection. Go right ahead. Okay, perfect. That looks like it's the majority of everyone. I'm now going to close the polls and let's take a look at the results. Okay, so in our audience, 0% said less than 100 EU milligrams, while 57% said less than 10, then 9% said less than 1, and then 35% said less than 0.5. So to get the right answer, Jebo, what is the correct answer for that?
Actually, we recommend less than 10 EU per mg. It's suitable for cell-based experiments because when you receive the proteins, you will dilute the protein during your experiment. So the final concentration of the endotoxin would be very low. Would be like some of the audience have selected lower than 1 EU per mg. I think, yeah, that's the answer.
Okay, perfect. Thank you. Let's jump to poll question number 2 now. So, audience members, this is our final poll question. So please go ahead and take a look at it and then make your selection from the options that we have below. And the question is, which of the following delivery methods can be used to deliver proteins for gene editing? Is it electroporation, lipid-mediated delivery, viral vector delivery, microinjection, or all of the above? So please go ahead and make your selection. And just like before, I'll leave it open for another 40 seconds just to get everyone involved in our final poll. And then we'll get ready to start the Q&A portion of the webinar. And just as a reminder to our audience, you still have the opportunity right now to send in your questions using that questions window, and we'll try to attend to your questions during the time we have together. Okay, this looks like it's the majority of our audience, so I'm now going to close the polls and let's take a look at the results. Okay, so here are the results. We have 7% that said electroporation, 3% lipid-mediated delivery, 3% saying viral vector delivery, 0% from microinjection, while 87% of our audience said all of the above. So to find out the correct answer, I'm going to pass it back to Jabo. And Jabo, what is the correct answer?
I would choose the last one, E, for this one. And no one choose microinjection. That's a
No.
very interesting scenario. Actually, I have a client that had used our PE Max to inject into the embryos of the zebra fish, and it shows 8% pure editing efficiency.
Oh,
So yeah, so I think this result is comparable to the literature reported.
okay.
So yeah, I think I think I would choose E.
Okay.
And
Thank you so much for
there
that.
is a lot of space for our scientists to keep digging into the delivery system of
Uh-huh.
proteins. That's why we are providing more proteins to try to help or assist our customers to achieve their to achieve like better results to when they are trying to work on the delivery system.
Perfect. And now I'll invite you to put on your webcam now, and we're going to begin the Q&A portion of the webinar. So as I said earlier, please go ahead and make your selection by putting your questions through that questions window, and we'll try to attend to your questions during the time that we have together here. And as a reminder for those of you watching on LinkedIn Live events, feel free to post your questions in the comment section. Okay, so I've already received a bunch of questions that have come in, so let's just get right to it. This first question here, this audience member is wondering about, does GenScript provide any of these nucleases, base editors, or PEs?
Actually, yes. As I mentioned, we already providing some PE and even some Mat7 is not even Cas9 system. And we also have ABE8E that we can provide to the customer. And we have P2, Pmax, and P6, BCD, and Pmax without RSH. in our stock. So as I mentioned, we also have this trial size for the customer, which is 100 milligram and only for $100.
Okay, perfect. Thank you so much for that, Jabo. All right, let's go to our next question. This is a little lengthy. This audience member is asking, could you share any data or insights on the in vitro editing efficiency of prime editors? Specifically, do you have any quantitative results or examples that highlight how efficient prime editors are across different target sites, DNA sequences, or PEG RNA when tested in vitro? Jabo, what do you think?
That's a very good question.
Mm-hmm.
Actually, as you may notice, I already showed in vitro cleavage activity of Cas12a protein, which will create a double-strand break. It is really hard for us to detect the nick on a double-strand DNA and detect the transcription happen in the reaction. So I'm still working on that, and I don't know whether any of you have tested that in vitro. I will keep working on that, and if you have any good insights, please contact me, and maybe we can try to develop this method together. Because I didn't see any existing competitors have this assay at this moment.
Okay, perfect. Thank you for that, Jabo. All right, let's go to our next question here. This audience member is saying, from what I understand, the licensing rights for prime editors are held by Dr. David Liu's lab. If we were to order these enzymes from your company, would we need to obtain additional IP permissions or licenses to legally use them in our research or commercial applications? Your thoughts?
Yeah, that's a very good thought.
Yeah.
Actually, that is true that W's team had this IP of PE system, PE editors, and I have to say, there is information I need to tell all of you that David Liu is our GenScript supervisor. So our company is contacting David Liu's team, and we are going, and we are the first one actually going to license in the PE protein, a PE protein for the production.
Wow. Okay. Thank you.
And if
Go
you
ahead.
ordered this PE system, PE
Uh-huh.
protein from us, I think if you use it as a R&D or in the research stage, there won't be any IP problem for you. But if you want to commercialize it, I think you need to get permission from the W lab.
Okay, perfect. Thank you. Let's go to the next question here. I think we have time for a couple more questions. What is the process for ordering trial samples of these prime editing enzymes? And additionally, then they have another question. Are there any recommended guidelines for handling or storing these samples to ensure optimal performance in our experiments? Jabo?
If you are if you are interested in ordering our proteins, you can contact me.
Okay.
You will see my email address, and you can send email to me, and we will arrange the shipment to you. Besides that, we can also provide customized sequence for you if you wanna try some your own design or engineered Cas protein, not only base editor or prime editor, include even Cas9 or Cas12, Cas13. It's okay. We can also provide this customized service to you. And for the shipping and the storage of this protein, we will ship this protein in dry ice, and I will recommend to store this protein under minus 80 Celsius degree.
Okay, perfect. Thank you. Let's jump to the next question here. This audience member is saying, "Oh, from your web, I could not find the link for the PEG. Could you share me with? Can you share the link with the product link?"
Uh, of course.
Hmm.
Um, yeah, but actually at this moment, this PE protein is not, um, cat catalog
Oh, okay.
products,
Hmm.
because we are still negotiating with the WS lab, so it
Hmm.
will take us some time.
Okay.
But if you want some of this protein, you can contact me, and we will send some of our stock to you.
Okay, perfect. Thank you for that. Okay, let's try to see if we can get two more questions in here. Here's another one. Is it possible to order PEG RNA and sgRNA and the prime editor protein together as a complete package? And hold on. And if so, do you offer customizable options to tailor the RNA and protein components to specific editing targets? They're being very specific with their question.
Yes, yes. And actually, GeneScript is not only providing proteins, we are also very strong at gene synthesis and peptide synthesis and antibody productions and a lot of area. So we can provide a one-stop solution for our customer
Oh,
from the design of pegRNA, sgRNA, and to production to the protein that you need. So if you're interested in this package or this one-stop solution, you can also contact us, and we will give you our professional advice.
mhm. Perfect. Thank you. Okay, this looks like it's our last question here. This audience member is asking, in addition to Prime editors, do you also offer other gene editing tools such as Cas9, dCas9 base editors and cytosine base editors? And if so, could you provide information on the specific types of versions that are available?
Yes, we provide sorry, we provide Cas9, dCas9, and different kind of catalog Cas9 products. You can find it on our website. We can also provide um, uh, AB8E and PE series of the proteins. And we are developing CBE at this moment. I think we will
Oh,
have some progress next week. So
oh, wow.
if you are interested in these enzymes, you can contact us, and we will try to find out what we can do for you.
Okay, perfect. Thank you so much for answering all those questions. We have reached the end of the Q&A portion of the webinar. And if we couldn't attend to your questions, the team at GenScript will follow up with you after this presentation. And if you have any further questions, oh, hold on here. Here's the email address, protein@genscript.com. And before I stay on that page, I just wanted to mention that if you're interested in hearing more from GenScript, please save the date for their next webinar, Cilia Proteins as Biomarkers of Vascular Health, scheduled for Friday, December the 13th. So stay tuned for when those links are available to register. So thank you everyone for participating in today's webinar. Here is the email address. If you have any further questions, please utilize this email address. And you will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, give me a few moments. Additionally, I will be sharing a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. And for those of you watching on LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, please join us in thanking our speaker for her time here today. So thank you, Jabo. We really appreciate your time. Oh, and don't forget, get more out of today's presentation by downloading the supporting materials that we have available under the Handouts tab. And we have two there for you. We have a PDF of today's presentation and also a flyer on GenScript CRISPR solutions. So go ahead and check that out. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X Talks, I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Bye, Jabo. Bye, everyone.
Bye. Good day.